Okay, good evening. Here I am again. Actually, I decided now not to do reactions, so not to do another Blackboard talk, but I uploaded my lecture notes, instead of including the first lecture, the second lecture, and the reaction lectures on the website of the program. So whenever you would like to discuss with me about these things, which are there, in particular kinetic proofreading is very interesting, also the allosteric switch model, the MWC, Monod-Weyman-Changeux model for a receptor, with several binding sites, interesting. So please just approach me, and then we can discuss this. I decided just now to present a bit of my own research, namely biophysics of killing. Once I suggested this title to a conference organizer, he told me it's fine as long as you do not do experiments. Yeah. So this is essentially immuno-biophysics, and I give you a very rapid introduction into immunology, and then talk about some physical aspects of that process. So what you see here are killer cells in action. The killer cells are actually the ones which are not colored, yeah? And they search for targets which are greenly labeled, and then they migrate through the environment, yeah, and attach to them, yeah, form a synapse, what is called the immunological synapse, and then kill them. That these are killed, that the targets are killed, you see, yeah, there is a sort of an apoptosis marker. Essentially, it gets colorless as soon as the cell is dead. Yeah. So here, maybe you see it again. It attaches first and does something which we will explore later. It kills it, detaches, migrates, and then the next cell. You see already in the experiment, of course, so that you can observe many killing events of these kinds. It's very dense with targets. This is usually not the case. This is where search problems come into play. Okay, what are natural killers or natural killer cells and T cells? Essentially, they have been called granulocytes, simply because when you look under the microscope, so in ancient times, what you have seen is that they have dots in them, so-called granules. And these we will later see are the vesicles which contain the cytotoxic materials, so very characteristic, yeah, for killer cells and T cells. What is in the killer cells, the NK cells and the T cells, they kill in very similar, in the same fashion. The difference is essentially that the killer cells are part of the innate immune system. Yeah. They do not have an antigen-specific receptor. They sort of genetically learned to recognize and kill some abnormal cells. But if something like COVID comes, yeah, then or Corona, then you need those here, the T cells. That is, they are part of the adaptive immune system. They have a specific immune response against any foreign antigen. Okay, and both kill pathogenic cells via the release of cytotoxins, so-called lytic granules. Yeah, these black dots that you see here in the. Okay, a killing process, including search and activation and so on, is a complex thing. So it involves first the problem how to discriminate between friend or foe. So that is a very I was fascinating when I learned how that is done. Then a T cell has to become activated. Why? I will tell you. So they are not sort of, when you are, they are not permanent, they're tons of T cells in your body, but nearly none of them is activated except once you get infected with a pathogen or you have a tumor inside or something. Then when they are activated, they have to search for their target. When they make contact with the target and recognize it, involving the friend or foe mechanism, they have to build a immunological synapse. The cell has to become polarized, as we will see, and then the shooting has to take place. That is the secretion of toxin. And then I don't mention this. There are immune evasion which countermeasures. So first of all, if that all works, if a T cell is able to recognize a cancer cell, I mean, there is no cancer problem anymore. So there, one characteristic aspect of cancer is that it can evade the immune answer. And now the current research is essentially device inventing methods to counter these immune evasions of cancer cells. Okay. Before I proceed, what you have seen in this movie is actually really scary. These T cells are serial killers once activated, and they kill everything. Once they recognize the cognate antigen on this surface. If that is if there is something going wrong, that is extremely dangerous for you. I mean, they are really potent killers, and they will kill you. And that is the problem of autoimmune diseases. So insofar, there are also many control mechanisms involved before the deadly shot is actually executed. Okay, the basics. You find, I mean, this is nearly as thick as the book Molecular Biology of, I forgot the 4 or 6 names nowadays, with over 1,000 pages. Alberts, yeah, Alberts and Bray and so on. It's not as thick as this because, and you can download it, at least the 8th edition of it. So I show some pictures of that. Janeway Immuno-Biology. So the basic facts is essentially this is part of the activation process. It's essentially dendritic cells, which are not the killer cells. Dendritic cells, they reside in the peripheral tissues and essentially collect garbage and material. So, which then they can present in the lymph nodes to inactive T cells. And from time to time, they find something which doesn't belong to the body. It's a sort of a short peptide, which is presented sort of on the, which is presented by the by a host organism, by a virus-infected cell or by a tumor cell also. So then they endocytose this, make it shorter, then present it on a machinery which is called MHC, major histocompatibility complex, and show it on the exterior. With that, they go to the lymph nodes, and they present this piece of foreign material, this peptide, which nowhere in your body occurs, and there will be some T cell which can actually recognize that. They get activated, they proliferate, and then they migrate out of the lymph node, and distribute over, for instance, via the blood vessel network over the body and look then for the pathogen. A lymph node typically looks like this, and sort of this is also something where dendritic cells as well as T cells have to navigate. Okay, so the point is that a single progenitor cell that can give rise to a large number of different lymphocytes, each with a different specificity. Specificity concretely means with different receptors on it. These receptors are not genetically coded, like, yeah, so that they have a stochastic components in them. I will show you later. Yeah. So then what essentially happens is from a progenitor cell, you produce lots of lymphocytes. Yeah, they can lots of different lymphocytes, meaning these different T cell receptors, which are able to recognize yeah, peptides can be produced. But some of them, yeah, they have then receptors which are essentially yeah, would like to bind to, can have a high affinity to peptides that occur in your body. So they get removed first, and then you have a pool of mature naive lymphocytes. And once they get sort of these naive lymphocytes get activated, they proliferate. And the thing is, yeah, what makes the specificity of the lymphocytes is its T cell receptor, yeah, which contains different parts, yeah, and which is essentially produced in a way like this. There are the so-called inherited gene segments, and there is a unique combination of these segments. They become sort of joined together by a somatic gene rearrangement. So essentially, there are gene segments which sort of, when a lymphocyte, a T cell is produced, they are stochastically reshuffled. And that then produces against things, yeah, peptides, for instance, or sequences, yeah, sort of that or affinities for sequences which do not occur in your body. It's essentially the idea. I mean, I was very fascinated. I mean, you must recognize something, which you do not know. I mean, a T cell doesn't have sort of a memory of all possible peptides that occur in your body. It also doesn't have any eyes to recognize or a brain to process it. So what is the solution to this problem? Everything in biology is yeah, most of it is, it's chemistry. So essentially binding of two proteins. Here in this case, the receptor and the ligand. So how do you do this? Yeah, the solution to this problem is simply, yeah, when you think of this key lock principle of biochemical binding, so essentially you generate a huge variation of random keys, yeah, and throw away all these keys which open locks in your own body. So then automatically you have keys that can open only foreign locks, yeah, which you have never seen before. And that is essentially what is happening in this somatic gene rearrangement. The molecular mechanism, yeah, if you're interested in, it's so essentially this is this particular gene, inherited gene sequence. It has so those segments which are then shuffled in some way, rearranged DNA, and also another sequence here, and they give then one part of the T cell receptor, which make them essentially then affine to something which is completely, I mean, this here still could also be a receptor that has affinity for things in your, for peptides in your body. And that first, those have to be eliminated in the thymus. And also another interesting form. So there is statistics involved in it, because you're having this mechanism. You can compute the number of different sequences you can actually generate with that. This is of the order of 10 to the 18 or even 10 to the 19 different sequences. So essentially, that is a big topic in statistics, but also in statistical physics. And there is a really nice talk of Alexandra Walczak about exactly this this topic in YouTube. There's, to analyze the T cell receptor repertoire, so it is called, how large it is, how probable it is that you have high affinity to randomly generated antigen, and so on. All these topics, they are sort of current research, and she explains it really nicely in this YouTube video. Okay, and as I said, what then essentially the T cell receptor does? So there are this so-called major histocompatibility complex 1. That is a complex, protein complex that presents peptides derived from a foreign antigen, and those by binding it to this MIG on the cell surface. And that is essentially how dendritic cells present those peptides to T cells in the lymph nodes. So
what is the function of T cells are, and what is the difference in the function of natural killer cells and T cells?
The T cells, they look for foreign antigens. So with the mechanism that I have described, just sort of you generate these huge amount of keys which only fit to unknown blocks. NK cells, they have receptors that unlock known blocks. So they are inherited sort of information. So the I show you in the first slide. This is the innate NK cells. It's the innate immune system. It's part of the innate, and so they recognize and kill some abnormal cells which are known. So whereas the T cell is sort of the very, very flexible killer, and that is part of the adaptive immune system against foreign antigens. Okay.
This antigen presenting T cells are the T4 T cells, right? These are helper T cells.
What I mean, they are also no. It's what I mean here is the presentation of antigens to T cells in the lymph node. So the dendritic cells, they, as I said, they cruise around in the peripheric tissue and phagocyte material, and then sort of chop it into pieces, small peptides, and then they present it, so incorporate it into the MHC. And the MHC goes to the plasma membrane, and there they're presented to the exterior. And then T cells come and look whether their receptor fits to this peptide presented in the MHC, major histocompatibility. And when it does, When it fits, there is another control mechanism, then they get activated. And activation means, in this case, change in the genetic program. So therefore, the gene expression pattern has to be changed, because you do not, the normal inactive T cells that do not carry this cytotoxic material around all the time. That would be highly dangerous. So, but this genetic reprogram means they now start to produce this cytotoxic material. You start to proliferate, in contrast, so that you get a large pool of the right T cells, which are specific for the intruder, and so on. Okay, so far this. So now you all understood how this works. So remember, I mean, this how T cells get their specificity to foreign antigens. It is. Yes?
Foreign which are entering, all of them won't be identical. So how is this recognizing a process optimized? Like we are saying that the key fit into foreign locks, but all foreign locks will not be identical. So how can one key fit in different locks? Every different foreign locks.
Not one key. I mean, there are 10 to the 18 keys. Essentially, I mean, potential. I do not. I mean, the number of naive T cells I do not have in mind, but this is also a very large number, 10 to the 15. I don't know, but they all carry different stochastically generated keys. Yeah, and so all T cells have another key, essentially. Yeah? And therefore, activation means also proliferation. So then, when this T cell proliferates, then this activated T cell, then they have all the same key. And that then essentially. Okay, so target search. So now assume that the T cell has been activated, or you have a natural killer cell. They have to search for targets. But I just mentioned that target search is relevant also before activation, because there the T cell has to find dendritic cells, and check their surfaces for this MHC, to get activated or not. So that target search also happens for non-activated T cells in the lymph nodes. But now, more important is, once they get activated, of course, then they have to migrate, for instance, through the tissue and try to find target cells. Yeah? So this is what I mean. And that has been actually investigated, and it was most, in these papers, in very early papers, they looked for the interaction of T cells with dendritic cells in the lymph nodes, so, or they put dendritic cells into collagen, into an artificial collagen matrix, and then they studied essentially the pattern of movement. So what you see is here, here are the dendritic cell number 1, dendritic cell number 2, here number 3. T cell comes and checks essentially this dendritic cell out. Yeah, okay. Didn't have this activating factor, then migrates to the next one, migrates to the next, and so on and so on. So you clearly see that there are stops at dendritic cells, and then moves forward. And in tissue, T cells in tissue, there was a time when there was a debate whether T cells, when they are searching for targets, they do a random walk or a Levy flight. And those questions have been investigated with 2-photon microscopy, because you have to analyze 3-dimensional random motion, or 3-dimensional trajectories of cells in tissue. So you have to sort of at least to reach to some depth in order to be able to analyze to get the tracks So it is an essentially, and then that was even published pretty high in Science, and here the other paper in Nature, where here they came to the conclusion, this is the the distance, so that the square root of the mean, the square root of the mean square displacement, yeah, versus the square root of time. You see this is nearly a straight line. Consequently, they concluded this is a random walk. And 10 years later, others have done, I think, longer tracks. They plot the mean square displacement as a function of time. Yeah, so it's a different plot from here. Yeah, and then they find over nearly a decade, so be careful with your decades, that this is sort of, this is log-log plot. So these are sort of straight lines here with the slope 1.4. So that is then super diffusion. And that is an exponent that you can get from Levy flights, which is another model for anomalous diffusion. Yeah. So, but I think that this question is not particularly, although that these papers have been published highly, I don't think that this is sort of at the heart, at the focus of the problem. Essentially, but we will see later, the search process is influenced by many things. Also by bystander cells and things like this. So the characterization of whether the mean square displacement goes sort of linearly or superlinearly sort of is interesting, but I guess at the exponent would change that you observe that you can fit here over one decade will change as soon as you change the environment, change the parameters, and particular, it depends on the fact whether you have chemotactic factors involved and things like this. Okay, now here are experiments of my colleagues. They mixed essentially killers, which are invisible, with bystander, with target cells and bystander cells. So target cells are those which can be recognized by the T cells, and will be killed then. And the bystanders just ignore. They sort of look like obstacles, more or less, to T cells. And with these assays, by fluorescently label the target cells with a apoptosis sensible fluorescent label, you can easily measure the killing efficiency of this ensemble of killer cell, of T cells, simply because you get a certain light sort of intensity from a large number of target cells that are still alive. When you kill them, they don't shine anymore. Kill more, it gets darker and darker and darker. So you just study the light intensity as a function of T. That is then just the killing efficiency. That is shown here. So target lysis as a function of time. But they didn't count the number of green points, they just measure the light intensity, okay? And then here so you see that as a function of time in minutes, here, it first increases and then saturates somewhere. But the surprising observation was essentially that including the bystanders, increased the killing efficiency. So this is certainly, after what I have taught you this morning, something very unexpected. The bystanders are, would be the first idea, are like obstacles. And when you increase the obstacle number, so and therefore increase the filling fraction, I showed you the curve today in the morning as a function of the filling fraction, the effective large-scale diffusion constant decreases. When the diffusion constant decreases, mean first passage time goes up, right? And that means as a function of time, the killing efficiency should be smaller for higher bystander density, and the contrary is observed. Yeah. So you can sort of check that in a simple model. Yeah, what I told you essentially this morning was single particle processes, but you can also perform this task with many particles, many searchers, many killers, yeah, many targets, many obstacles, and indeed you observe something, yeah, which is exactly sort of, so this is essentially the number of of killed, so removed targets for different obstacle numbers. Asymptotically, all targets will be removed. Yeah, where the obstacles are really obstacles. Yeah? So you can define a half time when half of the targets have been removed, and then you see that it increases, that the half time increases with the number of obstacles. This is sort of what you would normally observe, what is not observed in the experiment. So what's on? So for physicists who believes in the predictive power of his imagination and his understanding, there is only one conclusion one can draw from that. It is that the bystanders are not obstacles. There must be accelerators. Yeah, sort of when they are sort of in their vicinity, would be able to let's say accelerate the T cells through some biochemical mechanism. Then this is the prediction of this model here. So there is a region, we define a region around each bystander, in which sort of the diffusion constant of the T cell is increased. So that is what I say, that it's acceleration. And when we simulate that, you see, yeah, again, the same curves here, but now the inverse tendency, namely that with the increasing number of bystanders, the half time drops. Yeah. So that would be then essentially the prediction of this image, okay, of your conclusion, they must be accelerators. Then we went back to our experimental colleagues and said, okay, this must be, and please check. And that is what they have done. They have then essentially looked closer to their movies, defined the region around each bystander, yeah, and actually observed exactly what I just said. That when you have sort of the mean velocity of the T cells is without bystanders, something about 6 micrometer per minute. And when there are bystanders, the velocity is increased in the vicinity of the bystanders. So that made us happy. And then one can only speculate about what actually happens, that the bystanders can actually accelerate in such a way T cells in their migration behavior. And it is known that these bystander cells, they produce H2O2, yeah, and by simply living, and so increase the H2O2 concentration in their surrounding. And it is known from other experiments that H2O2 can actually accelerate the migration behavior of T cells. So essentially, that is then the solution to this problem. Hmm? A stable? No, here in this model, it's circles, I mean disks. I don't think that the shape will make a big difference. I mean, when you look extreme shapes, when you distribute the target, let's say, elongated drastically and distributed, then yeah. I mean, but when you look at the movies, the targets are not that extreme. So they are not particularly elongated. They look all pretty round. Yeah.
Experiment, you said something about H2O to whatever, but in the model there is no such thing, right?
It is. I mean, there is no H2O2. There is just this phenomenological acceleration here, these red parts in a certain annulus around the diffusion constant. Yeah.
Is it that in some such space, which unnecessarily it would have searched, that have been taken away, so it has to search over a much less area. The searcher has to search over a much smaller volume of area.
Yeah. I mean, of course, you can make that easily. I mean, there is no. I mean, for a wise man, there is no necessity to perform a simulation of this problem because essentially the model has the same. I mean, has the same path realizations as the one without acceleration. So essentially, the space of paths, possible random paths, is the same. It's only that segments of these paths are passed in a shorter time because of the increased diffusion constant. And that means automatically that you should end up, when you have this increased diffusion constant here, that you end up with a smaller mean first passage time. We can talk later. I just suppose this is the only path that is realized. Yeah. Then here, this part here is done faster in red. That is essentially the argument. Okay, maybe we can discuss it later. Okay, that was search process. So there are many interesting aspects in this target search process. Now, suppose the T cell or the killer cell has found the target and it recognized their cognate antigen. Then what happens is, essentially, here you have a diagram. This is the antigen presenting cell. It presenting the antigen AG in the major histocompatibility complex here, and the T cell here in red docks, binds their T cell receptor to this peptide here, to this major MHC with this peptide. Simultaneously, so all, not only one, there are many bonds. Simultaneously, it makes a strong mechanical contact with integrin on the APC side and ICAM on the other side. And then there is also sort of here a control factor, CD28, binding to CD80. So recognition and part of the mechanics in strong mechanical bonding is happening here. And then what actually happens is that a signaling cascade is then set into action, leading to sort of what is called sort of a nuclear factor in antigen recognition, and that then essentially penetrates the nucleus and causes the nucleus to causes a change in the gene programming. I know, not totally, but now cytotoxins become expressed and become produced, and the cell proliferates. And this process here has been studied a lot in TIRF experiments. So essentially, you present the antigen on a on a artificial sort of membrane, lipid bilayer. You present it, and then you essentially put the T cell on top, and then you perform TIRF microscopy, with which you can essentially visualize very well the plane. Yeah. And then actually you observe that what happens is that the integrin binding sites T cell receptor bindings, they self-organize into spatial, into this spatial arrangement that you have here the T cell receptors, and here sort of the integrin and ICAN. And that can be nicely observed in TIRF, where you see here this annulus of this binding. Here even better. So that is essentially what the picture, what one has of this, that one in this construction where you here have strong binding in this periphery, you create here a chamber where you can then actually exocytose the cytotoxic material, including perforin and granzymes. The perforin makes hole into the antigen-presenting cells, and the granzyme is apoptosis-inducing factor, which then diffuse through the holes made into the interior of the target cell and induce apoptosis there. So that is how one thinks of it. Okay. Once this happens, here is already indicated that the centrosome comes close by. This is the chapter repolarization of T cells during synapse formation. That is also has been mentioned by Raja in his talk, that this centrosome repositioning also takes place here. For which reason? Probably simply because the vesicles, so these granules, which one sees very prominently under the microscope, they are close to the centrosome. And then sort of you bring essentially the ammunition with the centrosome close to the synapse. So that is what's happening. Dynamics that you can see very nicely here in this movie, where you have in orange the target cell, in red essentially you find here in this non-labeled cell a red dot. That these are the NK cells which are labeled with lyso trackers. So lysosomes essentially are then labeled in red. They are typically accumulated, as I mentioned, at the centrosome. And when you observe this movie, you see that inside the cell, this red dot moves then towards the synapse. Yeah? And then when this happens, the poor target cell gets killed. Bloop. And here this fades away. Yeah. So this is simply an indication of before the killing is actually performed, this red dot goes to the contact area. Yeah. So what in the picture that people have of this process is essentially the T cell or the NK cells makes contact, yeah, with the target cell. Yeah. Then there's actin depletion. So actin is this here, this gray material here at the center. Yeah, after already 1 minute, so when you look at under the microscope, there is a lot of action going on mechanically. Yeah, it's really quickly deforming. It's really very similar to phagocytosis, but not complete, because killer cells are very, very small. And sort of it goes like this and then stops. And then you have retrograde flow, yeah, keeping of the actin, keeping sort of the shape. And but then very fast already. Then the sort of granule, they cluster at the centrosome, and then the centrosome moves, yeah, to this to the contact zone. And then deadly shot is done. Target is dead. Yeah, sort of repolarization takes place within minutes. Yeah. This is just an approximate number. It's not precisely like the Deutsche Bahn or so. Well, this is not a good example. Japanese Bahn, Japanese railway. It's which are right on time. It's not that. It's simply just the scale which one can estimate. And then target is dead, and then the NK cell releases the contact and goes away, which is also an interesting problem. How does the NK cell know that the target cell is dead? This is another point which is interesting to discuss. Okay, very early already in 22, it was sort of in this bright field microscopy video, it was shown that the internal structure, which is mainly cytoskeleton with organelles attached to it, actually moves prominently when a T cell attaches to a target cell during killing. So that is essentially what is going on. So the blue line represents sort of the path of what is assumed to be, let me show you again, assumed to be the center of the structure, so meaning the centrosome. Yeah. And it happens even when the T cell is attached to, it makes two synapses, yeah, with two targets, then it goes forth and back. Yeah. Once it goes here, whoop, it releases, whoop, it goes, yeah. So that is, that gives you already an idea what's going on. Yeah. Before I proceed to the mechanism of that and better experiments visualizing the process here, just want to mention, before anything can happen here, that any centrosome repositioning can happen, the microtubules have to attach at the right spot, like in mitosis. And then there they have to be pulled by dynein. Yeah, and this is the paper that already Raja mentioned, yeah, where you essentially, yeah, stochastically, the centrosome by dynamic microtubules stochastically search for the center of the immunological synapse. And then you get this prominent curve as a function of the angular position of the mTOR. Important here that has the right scale, time scale. Yeah. The time scale you can actually estimate already from just the dimensions of the problem and from the speed with which microtubules grow. Yeah, essentially, when you believe this picture of search and capture, then essentially the centrosome nucleates stochastically microtubules which grow in some random direction. So in on this within the sphere. That means they can either grow until they hit sort of the membrane and go around like this, or hit the nucleus and go around like this. The largest, sort of the longest path they have to go until sort of they don't realize until they, for an unsuccessful event is just go around, and typically then they start to shrink again. So essentially you shoot in random directions the microtubules, and there is a range of directions within which of angles within which you will find the target. And with that information and the radius of the cell and the size of the target, so essentially the pi times the radius, that is this path here, the maximum path, and the target size, typically a micrometer or 2 micrometers, and the gross velocity, it gives you an estimate for one trial time. So it grows and then shrinks again. For shrink and grows. So that with that trial time, that means it gives you, and then the estimate of the capture probability is just the ratio between the target ratio, and sort of the perimeter of this. That is a meridian, or no, that's not a meridian, or on the globe. These lines are called how? Longitude. And this one? Latitude. Actually, this sort of the circle at this latitude here, that has a sinus here. Means, of course, here is when the target or the IS, the synapse is here, the perimeter is smaller, therefore the hitting probability sort of is correspondingly larger. So that means essentially with that information, so the maximum, so the minimum ratio between the size of the target and the area which you can possibly hit, the segment, length of the segment which you can potentially hit, that gives you then essentially the number of trials that you on average have to do in order to hit it with a probability of order 1. And that gives you then with this trial time, the capture time. For one microtubule, it's smaller than an hour. Roughly. Means you have to try 30 times. On average, then you will hit it. And one trial costs 2 minutes, so 60 minutes. But you do not have only one microtubule. You have, let's say, 30 of them. And then you end up with 2 minutes until you hit the target. And then it is search and capture. Then it is then the microtubule is captured by dyneins, which are accumulating in the immunological synapse, and then the mechanics starts. And the mechanics has been, you got a glimpse already from the first video I showed, but here this very nice experiment by John Hammer. He was essentially able to manipulate the target cells with a laser pipette, an optical tweezer, and move them towards the T cell in such a way that they touch the T cell opposite to the position of the of the centrosome. Why? Then you can really see how the repolarization process most prominently. This was a very difficult experiment. As I said, most experiments in the formation concerning the formation of the immunological synapse are performed with these in these with these artificial membranes and turf microscopy. But then the T cell is very flat and sort of very broadly sort of distributed over the substrate. But this is not what happens. They stay spherical. In order to observe this repolarization completely, you have to do exactly like this. And this is then what happens here. The yellow triangle is the synapse, the contact zone, center of the contact zone. And the green dot is the position of the centrosome in the T cell. There you see how it moves towards the synapse. And now, this is the most brilliant part. Actually, they labeled the microtubules. Yeah? They're very, very similar to what is done in mitosis, as Raja discussed. And there you see what happens. Here, the yellow triangle is again the center of the immunological synapse. And this here is the centrosome, in the sort of the microtubules emanating from it in this way. You see that the point is that the T cell is pretty tiny. They are potent killers, yeah, but it's small. So in diameter, typically 10 micrometers, 12 micrometers, no more, and they have a large nucleus. So therefore, just by mechanics, sort of the microtubules, they have to go like this, pretty blended. And then what happens here is that at some point, a microtubule is caught here and then straightened. And it's not only one microtubule, it's getting thicker. There are several microtubules which are caught. And then the dyneins pull and with power on this string of microtubules, and the centrosome starts to move. And funny thing is that apparently, here you see you have 3 microtubules being apparently attached to the immunological synapse. And then when dynein exerts forces, they clap together complete due to mass action. And then when dynein pulls here, it apparently pulls against a wall, the membrane, and the microtubules depolymerize here. That is also known. The depolymerization rate of microtubules is force dependent. Actually, you can pull. Is that the next? Yeah, you can pull. There's this experiment by Lahn. File paper of 2012, where they decorate a barrier here with dyneins, and put here the centrosome with microtubules on this plate here. And when a microtubule actually catches a dynein catches a microtubule, then indeed the head of the microtubule is pulled against this barrier and gets shorter and depolymerized. Okay, so this is what these people here called the end-on capture shrinkage mechanism, for pulling. For dynein pulling on microtubules. There's also another mechanism that is then cortical sliding. Of course, it could also be that some dyneins catch the microtubule tangentially, in this way, and then sort of then pull in the same way, but the microtubule is then not necessarily depolymerized, but sort of the microtubule slides along the cortex. So there are 2 mechanisms. There are many more. Other forces are possibly involved in centrosome positioning. Yeah, but like for instance, growing microtubule in a geometry like this, they will then here push against this plasma membrane, and exert forces in this direction, which is in the geometry that I've shown you in this three-dimensional sphere not relevant, because all microtubules go along here. And then, of course, there are other mechanisms involved, but they do not play a major role here. The major player in the centrosome repositioning here is definitely dynein pulling on the microtubules, either in capture shrinkage mechanism kind or in cortical sliding. I skip sort of the quantitative description of semi-flexible filaments. Just emphasize that you have these two different mechanisms. And when you have here the synapse and the microtubules come in tangentially, then this sort of gives this kind of mechanism pulling on the centrosome, and here the capture shrinkage mechanism by which also the microtubules get depolarized. So we invented a model for that. So that is sort of what comes after the search and capture part, namely now the microtubules are attached, and they are represented by semi-flexible filaments in external force potentials which represent the membrane, so repulsive membrane, essentially, and the nucleus. You see the nucleus is pretty large, so there is not much space for the microtubules that forces this kind of morphology. And then the two different mechanisms, they are indicated essentially here. Cortical sliding is modeled by sort of here forces pulling microtubules tangentially along the membrane, and capture shrinkage sort of in this way towards the synapse, the center of the synapse, where it also depolymerizes. So this is also another back on the envelope calculation with which you can, on the basis of this model, estimate the time it needs to relocate the centrosome. Essentially, it's a balance between the drag force of this big thing. It's the whole cytoskeleton, microtubule cytoskeleton, with all the microtubules, but not only them, because there are organelles attached to the microtubules, mostly close to the centrosome, and that whole thing has to be pulled through marmalade, essentially the true viscous material. So you can estimate the time it needs simply by balancing this drag force. That sort of this the cytoskeleton, the whole cytoskeleton structure experiences when dragged with a velocity v through a viscous material. And on the other hand, the forces that are exerted on the microtubules by a certain number of attached dyneins. When you assume the force of the stall force of a dynein of 1 piconewton, could be more than that, at a certain number of microtubules, then you end up with a velocity with which the whole object is pulled through the viscous material between 4 and 18 micrometer per minute. And then the maximum distance is again this 15 micrometer. The radius of the cell is roughly 5 micrometer times pi is this 15. Before, so it's 1 to 4 minutes. But it depends, of course, it varies. Here, for instance, put 6, yeah, but here only 30, for instance, then then you need half that time. So it depends in this way from the numbers. And of course, nobody has ever counted the number of dyneins which are actually attached and so on and so on. So this is the how you estimate it. So then the simulation of the actual model you see here. So here you see how the capture shrinkage mechanism, how the a string of attached microtubule evolves, and then the whole structure finally evolves in this way. And here you see a thing that you also observe from a side view, more or less, that the microtubules, the microtubule, let's say, fence, so opens up here. That is also observed in the experiment. So it cannot be something mysterious, which is sort of biologically programmed. It's pure mechanics, because you see, you pull the whole structure. I mean, and then they are not fixed here, or only slightly fixed at the centrosome. You pull in this direction through a viscous fluid. And the viscous forces is essentially then push these microtubules to the side. Yeah. So you must see that the whole structure moves in this way, yeah, so that this microtubule here moves in this direction and in this direction, yeah, or it would be forced to move, but there is resistance. So it's yeah, this goes sort of, this here is going faster than this, and then meaning is pulled over. And then you can, as a function of the dynein density at the IIS, you can estimate, you can calculate how the distance essentially changes with time, and with that, and also the velocities. And with that, you see that the the time scales depend when complete relocation, when the relocation is completed, depend, of course, on the density of the dynein at the but when you assume, let's say, reasonable numbers, you see here, this is in seconds, then you are in the range that is also observed in the experiments. Now, you see, when you look at the velocity, as a function of the distance from the IS, first, initially, the centrosome is very far away from the IS, and it's pulled. So there is sometimes even a peak. The velocity slows down, and then goes up, and then goes down again. How can you explain it? So essentially, experimentally, these two phases have been observed too. There is an initial third phase, which essentially, I mean, each phase has sort of its meaning. First, so you start with the centrosome close to the membrane, and there's some distance sort of to the nucleus. So, and then suddenly dyneins attached to microtubules here push in this direction. So the first thing that the centrosome will do, it will move downwards. And then it will hit the nucleus. It cannot go further. Then you are here. Boing. And then the centrosome starts to slide along the nucleus. And boing, you see, it's already happened. You don't see it. So that was this phase here, walk. And now it starts to slide. It then goes faster and faster and faster, and then here it slows down again. That is then what you observe here in approaching the. Okay, this is cortical sliding. This was capture shrinkage. In cortical sliding, everything goes if you would assume pure cortical sliding, everything goes much slower. So when you see here the time scales, they are much longer. And when you look at the movie, you see also why. Because in cortical sliding, many microtubules will attach tangent sort of to the immunological synapse. And ones that go this way and then the others going that way, they will sort of will be pulled in different directions. So there is a tug of war. And then stochastically at some point, one side will win and the centrosome slides along the nucleus in one direction. But this is the reason why it's much slower. But when you combine them both, the cortical sliding capture shrinkage combine, a funny thing happens. The two mechanisms, they work in synergy. Essentially, because the cortical sliding mechanism will sort of guide the microtubules and catch them a little bit outside, because the capture shrinkage dynein essentially sits in the center, very concentrated, but cortical sliding dyneins are distributed wildly over the immunological synapse. Yeah, many of them get caught, but then the dynein of cortical sliding will sort of lead the microtubules towards the central capture shrinkage dynein, and so more of them get actually caught in the center. And you see here with very modest dynein densities, you get very fast relocation times. Okay. Briefly, what happens here when you apply this model to a situation where you have two synapses, you have to modify the model slightly and also include dynamical microtubules growing, shrinking, because when the process takes a bit longer, of course, new microtubules have to grow, because those which were performing the capture shrinkage mechanism, they are gone. Yeah, they depolymerize and then they're gone away. But that will that doesn't happen, and they stay away if you do not include that into the model. So you have to include it, and the two synapses in an angle gamma here. And what is also an ingredient, because you observe oscillations in this movie here, or not oscillations, but stochastic switches between one side, one IS and the other IS. There must be a mechanism which leads to these kind of stochastic force and back. And that means somehow this one must get the opportunity to pull this here back. This is only possible when by some mechanism the dyneins, the sort of here detach again from the microtubules. And actually in capture shrinkage, you see that at the end, the microtubule is strongly banded. And that means there are bending forces which are exerted on the bound dynein. And at some point, the bending just comes too strong, so that this releases here. And then sort of they take over and pull the centrosome back. So that is the mechanism that you suppose, and this is essentially then how it works. What you observe then over time, that indeed the centrosome goes forth and back between this synapse and this synapse. Boing, 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 boing. And when you increase the dynein density, then these oscillations or switches or the transition frequency increases. So that is the transition frequency as a number of as a function of the dynein density. So here it goes back. Okay, and this does not happen when you have cortical sliding. Right? Simply because here it is so, because there is, stochastically it can happen that the 1 or 2 transitions occur, but the problem is you do not have this release mechanism that you have in capture shrinkage. So the dynein stays attached. In the end stage here, in most cases, the synapse, the centrosome just stays at one synapse. Okay. I just want to mention that centrosome repositioning is also relevant in B cells, and it is sort of relevant also in dendritic cells. Recently, a collaborator of mine, Kiermaier, Eva Kiermaier, she published a paper about the amplification of centrosomes in dendritic cells. Actually, it turns out that dendritic cells do not have only one centrosome, but sometimes 2, and in many cases 2, or even more. And the big question is, what is the function of that? See, during activation, the dendritic cell must also enable transport of some material towards the synapse. And for that reasons, microtubules have to go to these synapse in dendritic cells too. But the question is whether the centrosomes are distributed, if there are 2, do they have different positions, and actually are they relocated once a T cell makes contact with them or not. And it turns actually out that the situation is more like this, that they cluster. The centrosomes in dendritic cells, they cluster during, and that is, in our view, plausible, because a dendritic cell can have more than one T cell attached, which gets activated. And then it's a question whether you reach these points from a centrosome faster when centrosomes are distributed or sort of combined or clustered in the center or close to the center. And it's easy computation to show that this here is, of course, better than that. Simply because the mean distance that you can compute here for the from potential targets on the periphery is simply smaller than that here. So it makes sense that they cluster. Okay, there's also calcium dynamics involved during T cell activation. So, but I think I will skip this. This is sort of the part where calcium, in my work, sort of calcium diffusion comes into play. Sort of essentially, you have at the synapse, you have a calcium channel, the so-called crack channel, calcium release activated calcium release activated channel that opens when the endoplasmatic reticulum gets depleted, involving stim and ORAI and so on and so on. So that is the channel, and there is a pump. The PMCA pumps are also accumulated there. And what actually happens when during activation is that the calcium concentration stays high for half an hour or an hour, which is usually deadly for a cell. So everything in a cell is arranged in such a way that high calcium concentration are very quickly sort of decreased. Because why is calcium high calcium concentration so dangerous? It's simply because this is the second messenger, and nearly all proteins have, or many have calcium binding sites, which serve essentially as an allosteric switch. As soon as calcium binds, or several calcium binds, the shape of the protein changes so that it can perform a certain function, which it could not perform before, or cannot perform a function anymore, which it could perform before. And now suppose, so it works like a key, calcium, in order to either shut down a particular protein function or switch it on. And now suppose you have lots of that, and then suddenly all proteins will go crazy. So that is the reason why calcium concentration is very low, 100, less than 100 nanomolar. That is then 60 calciums per cubic micrometer. Now here, during activation, the calcium concentration is increased for half an hour or even more. So how can you sort of, can the T cell trick this whole machinery which lowers the calcium concentration regularly? Trick is simply that upon relocation of the centrosome, mitochondria will also be pulled, which are attached to microtubules, pulled towards the synapse. And mitochondria, sort of they work like a vacuum cleaner for calcium. They suck in calcium and then distribute it inside and expel it at some other region. So this is, and the PMC sort of, which essentially they catch the calcium and redistribute it in the cell before the PMCA pumps can actually spit it out, which are concentrated at the IS. This is this model, and it essentially predicts exactly what has been observed, namely that in the cytosol, mitochondria are actually able to increase the calcium concentration. Okay, the last, maybe this is where sort of the delivery of these vesicles containing the cytotoxic material is also sort of a narrow escape problem because simply these vesicles have to be delivered at the center of the immunological synapse, and you can sort of deal around with that. And that I have discussed this morning, so I skip it. And when a vesicle is released, then so when the content of one of these lytic granules is released, then these things will happen that I have described before. Perforin is spitted out, which makes holes into the cell. Here you have such an exocytosis event. Perforin is spitted out, makes holes into the membrane. Either this here is mixed already with granzyme, or you have another vesicle which only contains granzyme, which also opens up. This is diffusing around in the synaptic cleft. It diffuses through the hole into the target cell and activates then an apoptotic enzyme, which then induces apoptosis, kills it. Perforin. And that can actually be observed by my colleagues. And what they have observed is that sometimes the cell is already killed by perforin alone. Sometimes the target cell is killed by apoptosis. That you can see, you can discriminate. When the cell is sort of, this is pictures how the necrosis, apoptosis. When the cell is killed by a perforin, it really disintegrates. There are blebs occurring, and loses its structure. So that is a pretty fast kill. So when there are many holes, then you get a kill by a perforin. But if that is not the case, then you observe apoptosis. There's an apoptosis marker with which one can visualize or label this particular death, kind of death. Yeah? And sometimes you observe necrosis with caspase activity, meaning they are killed via perforin and granzyme. The big question is whether this is all accidental, or so when you observe, yeah, sometimes necrosis gemished, gemished means mixed, A means apoptosis, so that they observe killing sequences. Yeah, and the question is, what is it good for? And is it good to have a number of fast killers, which only kill necrotically, and then a bunch of slower killers, or what is the reason for that? And that this is actually something which is ongoing research. Okay, I skip that also, and just show you the summary, and I hope that I have given you just a glimpse at this fascinating story of immunobiophysics, and thank you very much for your attention. Dendritic cells, yes. No, but you think so? I don't think that this is a reason, because what is observed is actually that the centrosome are sort of, they cluster. So they do not distribute, they cluster. Yeah, I think the point is, I mean, what is the point to have several centrosomes? The point is probably that you have a larger number of microtubules. That's it. Yeah, sort of, so that you can, yeah, sort of deliver, yeah, material to several synapses quickly. And these amplified centrosomes also play a role during migration. Actually, it is known that the positioning of the centrosome plays an important role in determining the direction of migration. Yeah? And it turns out that, that those amplified dendritic cells are also very quick migrators, sort of, which makes sense because they have to migrate around the lymph nodes and so on and so on. The amplified centrosomes, they migrate faster. Actually, what they do is their persistence is increased. And as I have taught you this morning, when you increase persistence length, yeah, your mean first passage time goes down. And that means that your activation efficiency goes up, essentially. Activation efficiency means simply the mean first passage time to find a T cell that can be activated. So this is the reason, I think.
So the two types of sliding that you mentioned, one was cortical sliding and the other one was like from the
Shrinkage.
yeah. So like can we compare it to the gliding assays in vitro for the transport, like cargo transport in vitro gliding assays that is implemented? Because they're also like effectively like motors are tethered to the surfaces and the microtubules glide over them.
Yes.
So it was like kind of, I don't know, I'm just asking. So the dynein motors, are they like tethered to the surface and microtubules glide over them along with the MTOC, or is it like something else?
So essentially, you have, let's say, microtubule look like this, and then it goes tangentially. It hits it's not the membrane, it is the cortex. And and dynein is attached to cortex. And in there, essentially, dynein attaches, tries to walk in this direction, and by this it pulls. This is the only aspect I considered here, but of course it makes all, for transport of material to the periphery, it makes, of course, sense to have a structure like this in contrast to, for instance, something like this here. Because here you can sort of cover more points on the periphery than with this. But this is here more efficient in pulling. So, because microtubule have two functions, not only here sort of mechanically, like in this case, relocating the centrosome, but also for transport. And for that, sort of the different geometries are also important. I don't know whether this answers your question, but this is how I understood it.
Yeah, I mean, I still think it is similar to the gliding assay.
What did you say?
The gliding assay. It's a kind of a procedure for implementing the transport in the gliding assay. Yeah.
Means why does it glide? During growth or during being pulled?
No, it's no growth or anything. Like it's like a glass slide over which the motors are attached, and then microtubules are like sliding over them. It's
You mean
yeah, kind of, yeah.
you mean the gliding assays to motors are essentially attached, and then you throw in filaments and then you observe how they are relocated. Yeah, essentially it's the same here.
Yeah.
We see difference simply that here this filament is attached to a centrosome and then can relocate it. But otherwise, if you would put attach things to the filament, like cargo or so, representing a centrosome, then it's exactly this mechanism.
Yeah.
Several mechanisms of death that you have, or killing rather.
Yeah.
There's another one which I think you have not mentioned, that is the B-cell dependent, right? I mean the B-cell dependent pathway where the antibodies will be killing.
Yeah, definitely there are also other, there are, as I said, there are also other cells involved, and there are other ways of killing, even for NK cells and T cells, because T cells can release cytotoxic material, and NK cells, and not only directionally, but also just somewhere else. Yeah, sort of by simply secretion into the environment, which is very dangerous, but in particular circumstances this works. And B cells I also did not mention, but they are more than that. I find this simply that had a certain appeal to me, sort of the whole process that reminded me sort of a movie Matrix, so where Nero approaches this machine town, and then suddenly the weapons are brought into the positions, and this was essentially because you know how dangerous this material is, which they sort of then transport to this one particular place. So I talked mainly about this. Yeah.
So I am telling that. I mean, is there any like? I mean, I am telling that that is the number of cells that T cells can kill. Is it a particular number, or it can kill as many as possible?
This is a big question because the problem is simply that in the experiment, the cells usually die at some point. Sometimes only after, sometimes only after 4 hours. If you do it better after 24 hours, optimally, you would not have this artifact of sort of limiting the lifetime, and then you could decide this question. So in the experiment, as I have shown you these killing sequences, some had sort of a length 3, and then or 2, and then they stopped. Others had a length, they made 12 kills, and then they stopped.
And one thing also is like when they are killing the cells, the material which is there in it, the cytotoxic material, they are releasing it. So it may change the tension of the cell, of those T cells.
Did they both?
The tension, membrane tension of the T cells. can change because it is releasing the contents of it.
Yeah, the title that this is when you have very efficient killing, then like what you would like to have when you do, for instance, immunotherapy and it starts to kill tumor cells very efficiently, then there's also the danger of a cytotoxic shock, simply because this is material then of the dead cells simply toxic for the body. So that's also not good. When they are killed too efficiently, that will harm your body also. So, yeah?
In cortical sliding, do
Yeah.
you consider the binding and unbinding rates of the dyning to the microtubule?
Yes. So that is one part of the model that you sort of you always sort of in each time step you compute your forces which are executed on the dynein via the mechanical deformations of the microtubules, and then there is a certain probability which depends on this force with which you detach.
So when you consider the unbinding rates for that means when there were two contact sites, at that time also there was no means frequency of movement of MTOC was decreasing for cortical sliding. So if there were uneven number of dyneins, that should means the frequency should increase. If the frequency is decreasing, that means that most of the time the number of dyneins that are attached to the microtubule, they are remaining the same. That's why
During cortical sliding, I mean, when you just consider cortical sliding, you do not have many transitions, simply because there is this tug of war. I mean, as you say, there is a mechanism by which microtubules can detach, but detached microtubules can also attach. And when there's a large number of it on either side, sort of, there is always the same number of microtubules. So that then keeps. It's only when one synapse, when the centrosome goes to one synapse, then many are attached here, and only a few at the other hand. So that means it stays there forever.
That the cytoskeleton gives a homogeneous search strategy for the cells, for the particle.
Yeah, that was this idealized case. So the one that has been considered in the literature before was essentially you have a cytoskeleton which is disorganized.
when you talk,
and we considered a cytoskeleton which is more, which looks more like this, and then here.
So when we are talking about like bystander cells, when T cell is approaching to its target, so but there is presence of extracellular matrix which has intermediary filaments. So will have you taken in consideration while
Intermediate filaments?
In the extracellular matrix which adheres the cells?
No, we didn't take into account what's happening going.
Because when you have shown the path of the cell, the T cell is going to its target. When you were showing a
in the model.
Yeah, in the model.
Here, no. I mean that would mean that the diffusion the diffusion has to be modeled differently. This was we assume Brownian motion, but when you have obstacles from filaments or so then you have to modify this. But I'm sure that that this can also be modeled by an effective diffusion constant, so on large scales. So it is then fine, I think.
About this diffusion that we are talking, accelerated diffusion, you can hear me?
Say it again.
Accelerated diffusion.
Actual.
Accelerated diffusion. that we were just discussing. So, like Dr. Debashish also mentioned, that when these bystander cells are there, you mentioned one case when there were obstacle cells, and the another case when you have bystander cells, and in the second case you see accelerated diffusion.
I assumed accelerated.
No, you also said that it has been observed in the movies that that the cells are moving in an accelerated manner. So let's say that your assumption is that there is accelerated diffusion.
Yes.
So I'm not questioning your assumption, but what I want to add is that the possibility for this accelerated diffusion, the mechanism behind this can be the volume exclusion that Dr. Debashish was talking about. Like when those bystander cells are there, they have taken up some volume, and if this cell which is diffusing through the medium is aware of their presence, it can basically exclude exploring that region.
But how can it be?
You also mentioned that they are releasing H2O2. That can act as a signal. I'm saying that this is a possibility.
No, I think I must, I must insist that because this was the first idea that our collaborators had. Essentially, they said, okay, the bystanders increase the excluded volume, therefore you do not have that much volume which you have to look for, to check out, and therefore the search must become more efficient. That is what they said, and I said no. I essentially told them the story which I told you this morning. When you have diffusion and obstacles, what you observe is when you have one obstacle, yeah, then you see this excluded volume effect. When you have two, yeah, do you still see the effect that you can calculate, yeah, by simulation? You still see the effect.
You are talking about.
But then when you have sort of when the search volume is completely filled with obstacles, then diffusion becomes. I mean, you don't. I mean, how do you use this information that you know where you do not have to search? Because first you have to touch the obstacle in order to realize that there is an obstacle, but then in that moment you cannot use this information anymore. I mean, it could only. I mean, the only way in which you could, for instance, use this information as a T cell is that when you touch it, realize that this is an obstacle, this is not what I'm looking for, that you sort of crawl ballistically along the periphery and then get released at the other side, for instance.
I'll come back to the H2O2 thing again.
I think this discussion can proceed in person. Some of us can discuss with him, others need not be held up. So let's thank all the speakers, including Professor Heiko Rieger, since he has completed his 3 lectures. Big round of applause for. And